Hello everyone, welcome back to our channel. Today we have an important topic to discuss, how to register your Metagenome project along with its sample metadata in the Genome's online database, also known as GOLD. First, let's start by opening our web browser and navigating to the GOLD home page. Once there, you'll see a register button. Click on it to begin the registration process. Now you need to specify the type of project you want to register. In this example, we'll start by creating a sequencing project. Click on the arrow next to sequencing project to reveal more options. Since this is not a public NCBI genome or metagenome, we can click on the No button. Please note that users don't need to manually submit public genomes from NCBI. If you have one to add, all you have to do is go to our Help section, send us a quick note, and we will import it for you. Coming back to our metagenome, once you have clicked on Sequencing Project, you have the option to search for your study name if you want to use your existing study. If not, click the button to create a new study. Note that GOLD follows a standardized canonical naming system for microbiomes which applies to a metagenome study name. Ensure that your study name follows the GOLD metagenome naming protocol, which you can find in the Help tab under GOLD Standardized Metagenome Naming Document. While ensuring that your study name follows the GOLD naming protocol, also be sure to provide a short description and fill in the ecosystem information. If you have additional information, you can include that as well. You can click on Create New Study to proceed to the next step. Here you will see two options to choose from, biome and organism. For whole genome sequencing projects, an organism needs to be created, whereas for metagenome and metatranscriptome projects, you need to define a biosample. In our case, we're creating a metagenome project, so click on Biome option and proceed to creating your biosample. If you extracted both DNA and RNA from the same physical sample, you can use a single GOLD biosample for both projects. If that is the case, you can go to the search bar and search for your previous biosample. If your sample is from a host associated, a soil, an aquatic, or any other environment listed below, you have the option to select the appropriate package to get access to additional metadata fields. For this demonstration, since my project is from the Strawberry Creek sediment in Berkeley, I'll use the sediment package. In the interest of time, I won't talk about all these fields, but do make sure to fill in the required information indicated by the asterisks. If you have doubts, feel free to hover over the information tabs for help. Once you are done, click on Create New Biosample. Next, we'll create a sequencing project. Fill in the required fields just as we did with the biosample registration. Make sure to provide accurate information for your project. Click on the Create New Sequencing Project button to create a new sequencing project. If you intend to create an analysis project, you can proceed and submit it to IMG by clicking on Create Analysis Project. And that's it. Congratulations, you've successfully created your metagenome project in the Genome's online database. Thank you for watching. I hope this tutorial has been helpful. If you have any questions or need further assistance, please leave them in the comments below. Don't forget to like and subscribe for more informative content. Until next time, take care.